Cheers. Okay, thanks. Well, it's a pleasure to introduce Iftikharul Alam for his confirmation seminar. So Iftikharul also goes by Russell. Many of you know him as by Russell. And so Russell traveled to Bangladesh at the end of February to visit his family, and he's been unable to return to Australia since then. So he's now giving his confirmation seminar from Bangladesh. And we hope he can return soon, but it's still uncertain exactly when he'll be able to. Yeah, okay, well, take it away, Russell. Thank you.
Thank you, Lucas. Welcome, everyone. Today, I'm going to present my Confirmation of Candidature seminar. So I'll be talking on my project, which is the vulnerability assessment of eucalyptus from different climatic zones to future drought and heat waves. So in this presentation, I'm going to talk about the background, the significance of the study, the objectives, the methods I'm going to use, and the references and other things. So here you can see an example of drought-induced plant mortality in Amazonian rainforest. So in this background section, I'm going to explain about drought along with its causes, followed by drought effects on plants. And later I will explain about woody tissue photosynthesis, which is a carbon dioxide refixation process of plant and its role in delaying drought-induced plant mortality. Drought can be defined as a prolonged abnormality of dry period when the amount of available water is insufficient for use. In case of ecological studies, we can say that drought can be measured through the deficit of moisture in the soil. In recent times, we have been facing several droughts like Millennium Drought, California Drought, the Big Dry, of which extent haven't been seen in the last 400 years. In the picture, you can see some example of Mura Reservoir and dried up Lake Hume, which is from the effects of Millennium Drought in the southern part of Australia. So if we see the decile map, which is a map of rainfall average compared with the entire rainfall record from 1900. So in the for the northern wet season, the rainfall decile for last 20 years is shown here. Rainfall during the northern wet season has been very much above the average for last 20 years. But if we see the southern wet season, then you can see that rainfall has been very low over the part of southern Western Australia during April to November. And in parts of southern Australia, severe drought has been expected to continue for the future also in the recent time. Some areas across the northern part of Australia has received less than 40% of their annual rainfall during this period. Here you can see the temperature anomaly for the last century. Australia's climate has warmed just over 1 degree Celsius since 1910, leading to an increase in the frequency of extreme heat event within Australia. If we consider the oceans around Australia, those have been warmed by around 1 degree Celsius since 1910. And it's also causing some marine heat waves and some problems around the coastal areas. So let's see how plants uptake the water from the soil. As we all know that plants need water for maintaining all of its physiological processes. So water is uptaken from the soil because of the water potential gradient along different parts of the whole plant. And at first, the water is uptaken by the root cuticles by diffusion. Later, it's transported through the xylem and reaches to the leaf due to the upward pull from the leaf created by transpiration. So here you can see that how the water is going up from the soil to the leaf. But when there is prolonged drought, it causes tension within the plant hydraulic system, and cavitation occurs within the xylem, and the air bubble present in the water expanded, and it causes the failure of the vascular transportation system and causes rapid plant mortality. So when the drought starts, plants try to close their stomata. As you can see, if the stomata is open, water will go out through the stomata through transpiration. So when drought starts, plant closes their stomata. Some plants shed their leaves, and due to the closure of the stomata, osmotic pressure adjustment need to be done by the plant, which is done by accumulating some solutes in the plant leaf. Pressure within the plant hydraulic system continue to creating bubble and cavitation. As a result, severe loss of hydraulic system, hydraulic functioning occurs and causes leaf photosynthetic capacity damage. So when there is less water in the soil, it reduces plants' physiological activities and tends to show some physiological effect like increase in abscisic acid and solute accumulation within the leaf. On the other hand, the photosynthesis, stomatal conductance, protein synthesis, wall synthesis, and cell expansion seems to reduce as the soil water potential keep reducing over the time in drought. Let's consider about mesophyll conductance. In most of the photosynthesis and plant photosynthesis models, mesophyll conductance is not is ignored. So what is mesophyll conductance? You can see that carbon dioxide has to be diffused from the atmosphere to the substomatal cavities, and from there it has to diffuse further to the mesophyll chloroplastic positions. So if we consider about mesophyll conductance, it is the diffusion of photosynthesis carbon dioxide from substomatal cavities to the mesophyll. So in recent models, it has been ignored because of the complexity to measure mesophyll conductance, but it's an important feature for the plant. So when drought occurs, it changes the forest dynamics. Like in the tropical forest, when severe prolonged drought has been occurring for a long time, it leads to increased plant mortality, and it disperses the shade and drought tolerant savanna species. Here in the photo, you can see the mangrove of Kuramba. In case of mangrove, less salt tolerant species are replaced by more salt tolerant species. So if we consider the woody tissue photosynthesis, which is a plant mechanism done by the seedling, sapling, and most of the woody plants which have smooth bark, they have a green chlorophyllous tissue layer just below their bark. So in presence of sunlight, this photosynthesis on the woody tissue can help the plant to refix the respired CO2 in presence of stem cells. So when there is less water in the soil and plant closes their stomata, they cannot uptake the carbon dioxide from the atmosphere. During that time, this kind of woody tissue photosynthesis can help the plant to reduce the effect of drought, and it can help the plant in delaying the mortality. Cortical photosynthesis plays an important role to supply the locally available carbohydrate within the near the xylem parenchyma cell. As young plants have less carbon storage opportunity than the big plants, cortical photosynthesis might play an important role for the survival of the young plants, and it might help the plant to adapt during the prolonged drought event accompanied with heatwave events. So let's see about the significance of the study. So as we know that about 800 plant species from 3 genera, Eucalyptus, Corymbia, and Zophora, is known as eucalypts. Here you can see the distribution of Australian forest types. Most of the Australian landscape is dominated by eucalypts, which is almost 77% of the total native forest of Australia is somehow some kind of eucalypts. So this is the distribution of eucalypts native forest in Australia. Eucalypts have evolved from their rainforest ancestors, becoming especially adapted to drought and nutrient poor soil, and they are adapted for frequent fire also. So despite their wide distribution all over Australia, recent researchers have found that many eucalyptus are found within a very narrow bioclimatic zone. So it can be said that more than 20% of all of the eucalypt species have a distribution range that extend across less than 1 degree Celsius. So in this study, we will be trying to look into some new insight, which are the physiological mechanism of eucalypts used to cope with drought and heat stress, and how this mechanism vary among eucalyptus species from different climatic region. We hope such new insight can be incorporated with land surface model to improve their predictive capability and in respect to climate change impact on important component of this eucalypt, which is an important Australian flora. Recent studies have found that inclusion of plant hydraulic model into land surface model can result in significant improvement of predicting the drought-induced hydraulic failure for different species. So the result from this project would be very much valuable to constrain the model prediction and develop the predictive capacity of the model. Keeping that in mind, the main objective of our study are we want to look at if there are any coordinated traits which can be used to predict the resistance to drought for eucalypts, how refixation of respired CO2 within the eucalypts play an key role in delaying their mortality during drought, and does mesophyll conductance in eucalypts decrease in response to drought? So for that, we'll be doing 2 different climate shift common garden experiment in Cairns in dry season and in Canberra at Australian National University in the summer. It will be done over the research period. So the targeted species and their dispersion over the different climatic zone in Australia are shown here. As you can see, we have selected species which are dispersed over different climatic region, so that can be a good representative for our study. So for the experimental design, we'll be taking 12 uniform seedlings from each species, and they will be well watered throughout the experiment, and they will be used as a control. 12 additional seedlings from each species will be used for drought and heatwave experiment. We will induce drought in 2 phases. At the first phase, the plants will be dried until there is any visual wilting, and then they will be rewatered for 10 days to allow them for full recovery. And then we will start our second drought phase experiment. During that time, water will be completely withheld to allow the plant to use all of their available water reserve. For the heatwave experiment, we will take half of the seedlings from each species from the drought experiment to the heatwave experiment after 4 weeks of the beginning of the drought experiment. So in the heatwave experiment, we'll increase the heat from 6 degrees from the ambient temperature. So for measuring the hydraulic vulnerability in plant, we'll be using optical method, which will be done by a custom-made camera with the help of Raspberry Pi and some additional programming. Here we can see that the process of measuring embolism formation within the leaf. So it's a visual technique where we can use the photos and other things to see the embolism formation within the different parts of the plant. So for measuring leaf water potential, we'll be using pressure chamber, which is used through pressure bomb. Here you can see an example. For the optical method, we can prepare the branch by taking out the upper bark, and then we can set up a newly customized camera on the prepared branch, and on the other side of the branch we can use a psychrometer to continuously measure the hydraulic vulnerability of the plant, water potential of the plant. In the photo, figure 13, you can see that embolism formation in eucalyptus, which have been measured through the optical method. So some previous result done by researcher has been shown that this optical method goes very well with the very well-known cavitron process, and the results somehow shows the similar trend. And the result is discussed by comparing the photos and taking the differences within the photos, so we can identify the percentage loss of xylem function through comparing the photos. For measuring the woody tissue photosynthesis, we have to cover the whole stem so that there cannot be any woody tissue photosynthesis, and we have to compare with an uncovered plant. So for leaf gas exchange measurement, we'll be using a youngest fully expanded leaf and use the Lycor for the measurement. We'll be measuring photosynthesis, stomatal conductance, transpiration rate. We'll be doing morphological measurement like leaf morphological traits, LMA, LT, LD. We'll identify the stomatal structure and density through nail polish smear method. For measuring the mesophyll conductance, we'll be using tunable diode laser known as TDL connected with a Lycor, and it will be used to measure the carbon isotope discrimination at different phases. Later, these values will be used to calculate the mesophyll conductance. For a statistical analysis, we'll be doing full factorial mixed model analysis of variance, which is known as ANOVA, where main effects will be the species and the treatment will be the fixed effect. These are the reference I have used for my background studies also and for preparing these slides. Here you can see my thesis outline. This is the timeline of my study. So the first part of the study will be done in Cairns and the second part in Canberra. As you know that due to COVID-19 pandemic, right now I am stranded in outside of Australia, and I don't know when I can return to Australia. So this is hampering my project. So I hope scholarship and course extension might give some help to cope with this problem. Here, I would like to acknowledge James Cook University, Australian National University, and Australian Research Council for providing support for this project. And thank you everyone for your patience.
Okay, thanks very much, Russell. We'll just, okay, yeah, so I think we're set for the sound here. So perhaps I'll take questions first from the room that I'm sitting in. Any questions in the room? Alex has a question. So so if anyone, when someone asks a question here, I'll repeat it so that the people on Zoom can hear it.
So it's a question about the stem photosynthesis.
Okay.
Russell, what do you expect the trend to be between all the species of eucalypts you're looking at as to the importance of stem respiration, and how do you sort of envisage comparing them?
Okay, so Alex asked about stem photosynthesis and how you expect that to vary across the different eucalypts from different climate zones, and how might you make that comparison? How would you quantify that and compare among the different species to say it's more important for some species than others?
Thanks, Alex, for your question. My hypothesis for this cortical photosynthesis is that the plant species from the drier climatic region will have a much more efficient way of cortical photosynthesis. So they will be much more efficient in cortical photosynthesis than the tropical eucalypts. And to identify those, we are planning to expose some experiment where we'll be covering those species with aluminum foil so that some of the plant cannot do cortical photosynthesis and some of the control will be doing the same cortical photosynthesis, and we'll compare about how efficient they are within the species and across the species climatic zone.
Okay, great. Thank you, Russell. Any other questions from in the room here? Okay, so from the Zoom audience, do we have questions? Yes, Lynn.
Hi there, Russell. Thanks for your talk. I was wondering, you showed a picture of what happens when plants are exposed to drought, and you showed us measurements that other people had made on rosemary showing what happens when plants are exposed to drought. And so I'm wondering what it is about eucalypts that you think is going to be any different from all these other processes. Like, can't we just predict it? Why would you have to do this on eucalypts?
Thanks, Flynn. For eucalypts, actually, eucalypts, as we all know, that eucalypts are the main plant species in Australia, and it's very much important for the economic and some other uses, like making furnitures and other things. So for eucalypts, we need to know this drought effect on eucalypts, because most of the studies done haven't seen the mesoville conductance effect on eucalypts, and most of the land surface model and also the photosynthesis model has ignored the measurement of mesoville conductance, and they didn't include it. So if we know the mesoville conductance values and can find a better way to include in those models, and if we can identify the trait of the change within the behavior of mesophyll conductance within the eucalypts, then we hope it will help the present land surface model to better predict the behavior of the plant in drought and heat waves event. I hope this answers your question.
Yeah, I think so. I'll let other people have a go. I have a couple more questions, but
Okay.
other people first.
Okay, anyone else on Zoom have a question? You can feel free to chime in to unmute your microphone and to ask a question if anyone has a question now.
Russell, it's Patrick.
Hi, Patrick.
I wondered if it was useful for the audience to hear a little bit more about the parameters that you want to get and how they might fit with the modeling context. You know, what parameters are you most focused on for improving the models?
Okay. Thanks, Patrick. So the main parameters we'll be focusing on are like the stomatal conductance values and then mesophilic conductance values, and we'll be look at the P50 values, which is the percentage loss of 50% conductance within the xylem system. So by that, we can create the vulnerability curve for each species, and we can somehow predict at which point the plant will lose all of its xylem function, and maybe it can die through desiccation. And also for other study, other part of the study, like particular photosynthesis, we'll be looking at photosynthesis done by the plant, or which can be like hindered because of the cover we'll be putting, and we'll be looking at the water use efficiency of the plant, and also we'll be looking at the effect of like the vapor pressure deficit, which is the amount of water holding capacity of the atmosphere compared with the maximum capacity of the atmosphere to hold moisture. So we'll be looking at those parameters and try to include those in the present land surface models.
Thank you. Thanks a lot, Russell. It was a great presentation,
Thanks,
by the way.
Patrick. Thank you.
I have another question. It seems as though other people don't. I can't see anybody, so I don't know if people have their hands up or whatever. Is it okay if I ask another question?
Yeah,
Yeah,
sure.
definitely. Yep. Go ahead.
Okay, so you're stuck in Bangladesh. Are droughts a thing in Bangladesh? Like, is there a way you can do anything apart from just suspend your PhD? Is there some work you could do now, anything like that, that would allow you to make progress in the face of this problem?
Actually, not really. I don't have access to that kind of facility here. But I'm trying to develop my skills with the measurement techniques. So I'm working with the custom-made camera with Raspberry Pi and other things, and how I'll be using those and trying to figure out the function of those cameras, and also looking at the previous models where I can fit my data to improve the capability of the model. But as I have told, I don't have access to do the actual experiment here. I don't have access of that kind of facility.
Okay. What else did I want to ask you? I had another thing. Oh yeah, the distribution of the trees. You showed us a map of Australia with some green bits on it, and you said that's where eucalypts are. But then
Yeah.
when you showed us your actual trees that you're working on, a couple of them went right across the arid zone. And so it seemed as though the distribution of the trees was, unless there's a color there that I can't see or something, it looked to me as though the distribution of these trees were much more arid than was indicated by that map of the, I was confused about the map.
Okay, let me show you that to you again. Actually, there was two map. Yeah, it was my mistake. I didn't show it. So the first map is northern wet season. Here, you can see that
No,
most
no,
of the
it was
part.
one of the eucalypts.
Yeah, yeah. Yep.
So not
I'm going-
rainfall.
Okay. This
That
one.
one. Yeah, so that one you said that the eucalypts are really confined to a small change in temperature.
Yeah. Like the eucalyptus species, like suppose one eucalyptus species, like Eucalyptus regnans, it has a narrow distribution, like less than 1 degree Celsius.
Mhm.
So the distribution is not that much for some species, but for some species, they're like widely distributed. As you can see for,
So this here says the distribution of eucalypt native forest. Is that not, is that just one species, is it?
sorry, can you ask?
I'm sorry, I'm just, no one's asking questions, so I'm going to find out stuff I want to know. If you go back one again to your green slide, the one with green distribution on it. So what, is this just the distribution of one species of eucalypt or of all eucalypt?
No, this is the distribution of total almost 800 different species of eucalypts.
Okay, so when you go, so there's a big white spot in the middle, gigantic arid zone region with no eucalypts in it.
Yeah.
So if you go to your list of species, the next slide, yeah, you have your list of species and here are their distributions. And when you get to the Carimbia, they're right across the whole there. So they're right across the arid zone. So are you making a distinction between eucalypt in particular and Carimbia? Is that important to your study? I thought
We haven't thought.
eucalypts.
Yeah. Yeah, there are eucalypts, but we'll not try to distinguish between them, between Corymbia and eucalypts, but eucalyptus. This map, I have secured this map from Atlas of Living Australia. So they have records from maybe back 200 years back. So these are the occurrence of the species they have seen. So it might not fit with the actual present scenario of eucalypts.
Oh, I see. So they might be gone from the central area.
Yeah, maybe.
Okay. The other thing I was wondering, given the extremely wide distribution of these plants, what's the chance for geographic adaptation to different environments?
We have to think about that and try to incorporate our project.
So you're going to be sampling trees from different locations, for example?
Yeah, we'll be collecting our samples from like around Cairns for Cairns studies and around from southern part of Australia when we'll be doing our project in Canberra. So the plant samples will be collected from different sources.
But they're pretty wet. There's both of those are fairly wet, right? Cairns, in my experience, is fairly wet, and around Canberra, is Canberra considered wet? I don't actually know that.
During the summer, I think it's much more drier.
Okay. Okay. That's all I got.
Okay, well, I think given we've got kind of a slow start and it's 11:42 now, I think we'll wrap up the question part of that then, and so the supervisory committee plus the Lynn and Lori, we can continue on Zoom, and I guess that Russell will log out of Zoom at this stage, huh?
Okay.
Thank you, Russell. It was a really nice presentation.
Yeah, thanks, Russell. It was great.
Thanks,
Thank
Russell.
you. Thank you.
And thanks everyone in the room. You're now asked to depart.
So I'm leaving the meeting right now.
Okay, thanks, Russell. I'll give you a message when we finish the our part of the meeting.
Okay. Yeah, sure. Thank you.